So we'll spend the rest of this morning talking a little bit about writing and visualization, and then I'll do a little elevator pitch of the 6 topics, and we'll pick topics. Great. So Stephen, if you want to start.
Certainly, David. Thank you. So thank you all again. Welcome back from the break. You see we put plenty of break time in to try and break up the monotony of the Zoom experience. So the overarching rule of writing any piece of scientific literature is to write for your audience. Writing for a journal article has very different goals than writing for outreach and education. When you do write for outreach and education purposes, you need to use a minimum of jargon while still preserving factual accuracy. You need to focus on the big topics that are of general interest, not the specific details. So the people who are new to topics, the curious public, they want to understand the forest. They are not necessarily equipped to understand each of the trees or the leaves on the trees. So you've got to focus on the larger topics that are going to be of general interest. And one of the nice things about writing for outreach and education is that your style can be a little bit less rigid, less formal than when you write for a journal article. It can be more conversational, and it's an opportunity for you to find a different kind of voice in your writing. Next slide, please, David. So a few guidelines. Even when you're writing a formal scientific paper, you're telling a story. And in the context of outreach and education, science communication more broadly, you need to tell a story that fits into the reader's own life. So you need to endeavor to make it relevant for them. Brevity is the soul of wit, as is often said. You need to keep it short and keep it to the point. And you'll see when you look at various Molecule of the Month articles that have been published over the last 2 decades, that they are very, very streamlined and to the point. There aren't any extraneous words in those articles. And again, when you're telling the story, it's going to be about the big picture. Don't get lost in the details. But nonetheless, you do need to find connections and examples that put the molecule that you're featuring, the Molecule of the Month, into its larger context. So I think you could see how David did that with the SARS-CoV-2 main protease Molecule of the Month that he highlighted before the break. He explained that this enzyme was essential for the virus to be infectious, and that understanding its 3D structure was going to be important in identifying potential inhibitors that could be made into drugs to treat the infection. So don't get lost in the details, and make sure you connect the molecule, connect the story to something that the reader is going to be interested in. Next slide, please. So when you are writing for publication in PDB 101, you need to treat this as a professional authoring activity that's no different from writing a paper for a scientific journal. Exactly the same level of attention to detail needs to go into your writing. Whether you're writing for a general audience or writing for a specialist audience. You need to proofread carefully for spelling and grammar. The thing that saved me over the last decade or so is the spelling and grammar check in Microsoft Word, and you've absolutely just got to be careful. you need to ensure, I mean, and for two reasons. You need to ensure that you're telling the story as you want to tell it and not inadvertently using an incorrect word or using a grammatical construct that makes your message unclear. But the other reason for being very careful in the way we write as scientists is that the introduction of even simple errors into manuscripts will lead to people doubting the truth of the science. If there are typos, if there are you use incorrect words, etc., in your PowerPoint slides or in your papers, when your peers see those errors, one of the questions that naturally comes to mind is what I'm being told about the science true? So that's I view that as the principal reason for being so careful with the way that we write and the kind of slides that we use when we're giving scientific presentations. You want to be convincing, and to be convincing, you have to be accurate in all things, not simply the data slides that you might show as part of a presentation or include in a scientific manuscript. Next slide, please, David.
So I can take over here,
Great,
Steve.
thank you.
And I just want to underscore that comment about proofreading and accuracy, and that, I mean, this applies to the stuff you're going to do in this workshop today, but also everything you do in your career. When you start writing grant proposals and journal manuscripts, if your manuscript is full of errors, the reviewer's going to ding you on that, and they're going to question your science. So it's always good to take that extra day and do a careful proofread of everything that you send out from your lab. Good advice for your future career in science. Okay, so I'm going to just walk through a few examples from Molecule of the Month articles that exemplify some of those concepts that Stephen just talked about. So this idea of editing and reducing jargon, for those of you who are already writing scientific papers, this is going to be really, really hard, because we're used to writing a certain way in science, which generally has a lot of facts and a lot of details. And those facts and details in general are not relevant when you're doing these kind of outreach articles. So for instance, here's a sentence from the journal article that I used as a source for the coronavirus article. And you can see it has a lot of information, base numbers, open reading frames, number of open reading frames in the genome, stuff like that. None of this is going to be of interest to a high school student. They're interested in the fact that it's a long RNA strand, and if you can find little facts like this, that it's one of the largest, largest or smallest or most common or least common or whatever, those kind of facts are always great for these kind of outreach articles. Just getting to the very heart of the concept without all that extraneous detail. While at the same time having a reference somewhere that has this information to back up this fact of it being long and the longest. Okay. Whoops. Oh, come on. Let's go backwards. Really, I know how to use my thing. Here we go. So here's the second one, also from the coronavirus article, talking about where the viruses came from, full of jargony words, zoonotic, molecular scaffolds. Words like that are not going to read real well with these audiences. So I tried to write it in a way that's much more conversational, saying this is a challenge that the medical community has, and we do that by making drugs that block a wide collection of different viruses. And actually this one word that I left in it, progenitor, came from the reviewer of the article, suggested putting that back in. That's not a word that I would normally include in these kind of articles, but as a nod to the person who reviewed it, I put that back in. Again, trying to make it much more conversational instead of using a lot of very specific and useful scientific buzzwords, but not useful in the context of these audiences that don't have as deep a knowledge of the subject matter. Here are a few examples of those hook sentences that we use at the very beginning of the article to try to get people interested. And to me, this is half the fun of writing these articles, is trying to come up with one overarching concept that's going to unify the whole article and be the topic of the story that you tell through the article. So in cases where it's related to medicine, that hook is usually pretty easy. So in this one on hepatitis C virus, the hook is saying, if we know stuff about our enemy, we can fight it. And then scientific knowledge is being translated into clinical success. So that's how the article begins, saying that we can use our knowledge to take medicine forward. This one about adenylyl cyclase used an analogy to technology, to a signal amplifier, and then talked about how the enzyme works like things that we're familiar with in our real life, amplifiers and radios, etc. This one about capsaicin repressor was super easy. Have you ever wondered why a pepper tastes hot? The rest of the article is talking about why it is, or how we sense hot tastes, and how the receptor is important in that. And this last one, talking about coronavirus, started with this thing of these viruses don't bother with DNA at all. Everybody, of course, knows that DNA is centrally important to all of life. But this article could say, but for these, they don't even need it. And then you spend the rest of the article talking about why they don't need DNA, why they can do everything with just their RNA. Here are a few examples of within the article of trying to show context of things within people's own lives. So that same example of the peppers, why do peppers taste hot? That's something that people know from personal experience. And then the rest of the article is talking about how the receptor ends up having the sensing of heat, and hence the sensing of spicy food is done by the same receptor. So interesting topic, right? We understand what spicy food tastes like. This is why it tastes spicy and why it tastes hot. For the article on the signaling within plant cells, I spent a couple sentences just talking about why plants are resilient, why they can grow back from pieces, why dandelions are so hard to get rid of. And then later on in the article, after people are relating that to things they might see in their own garden, then talking about how signaling networks give plants those properties. Here's an example directly related to a disease state, multiple sclerosis. Those kind of things are usually very easy, right? Everybody's heard of multiple sclerosis. So then spending the rest of the article talking about how the molecule relates to that disease state. And then this final one was talking about the spikes on the surface of SARS virus. And I was able to dig up a reference that talked about how the flexibility of the spike, how it's more flexible than other coronaviruses that cause common colds, and saying as being a structural reason for why this SARS coronavirus is more virulent than the garden variety coronaviruses that cause common cold. It's an interesting thing, you know, everybody's interested now in why this particular coronavirus is more dangerous than the colds that we typically get. So those are the kind of stories that we're going to want to tell. So moving on to the visualization aspect, the guidelines are exactly the same as the guidelines and goals that we have when we're writing these things. we want each of these pictures to tell a story that brings each of these molecules to life. I usually think of the pictures as being a molecular portrait. So when people look at it, I want them to get a feeling of the shape and the size and just a little bit of the personality of the molecule. Not trying to tell too much about it, just one or two little factoids about it. In these pictures, we also want to do a lot of editing. You don't want to try to show too much in one picture. And that's something that we do in our journal figures all the time, is we throw everything in the kitchen sink in these figures. Whereas these illustrations for outreach, they really should only tell you one or two things, just so that you don't get lost in all of the details. And then, as with the writing, we want to try to look for a big picture. If you can tell kind of a story about it, the picture, that's always nice. And trying to reduce all of the jargon that we typically have in our pictures. Assuming that our viewers don't understand everything there is to be known about ribbon diagrams and space-filling diagrams and bond diagrams and everything, all that visual jargon makes for a very confusing picture. So instead, we want to come up with a simplified image that just gives, as I say, a portrait of the molecule. So we'll show some examples of that as we go. So for these main pictures, I've been using this illustrative technique for creating the pictures for many years, and we'll do a tutorial tomorrow on how to create those. And so they create a very simplified, familiar, friendly portrait of the molecule. And so this is just comparing the figure that from the journal article I used as the source for this article and the picture that I generated for the outreach article. You can see that this journal article is trying to show a lot of different things, right? All in one picture. It's trying to show the experimental electron density for the inhibitor, multiple views, ribbon diagrams, surface diagrams here. You can barely see the inhibitor here, unfortunately, which is the major part of the story. Lots of stuff going on in this picture. All of the residue names for residues that are interacting with the inhibitor. These are all super useful things in a journal article, but way too much detail for outreach, right? So people reading these outreach articles, they're never going to need to know that glycine 143 is over here. So you throw all that information away, distill it down to one portrait here that shows that the protein is a dimer of two identical subunits, and there's an inhibitor bound in the active site. That's the only thing this picture shows, and it's the only thing that the picture needs to show. I'm also a very strong advocate of this kind of space-filling representation that shows each atom as a sphere, because to me, they really nicely show the shape and form of these molecules. They also let people know that molecules are composed of atoms, because you can see individual spheres for each atom, whereas these kind of surface representations, it's harder for me to see that there are actually atoms in those. And yeah, so that relationship between molecules and atoms is clear. And the colors are chosen, again, to try to simplify that story that we're trying to tell, that there are two subunits here that are similar, and the inhibitor is a different molecule than the proteins. I typically use this very simple coloring scheme with carbon atoms in a slightly lighter shade than nitrogens and oxygens, just to focus attention on the whole molecule shape rather than all the little atomic details. But it's still there if you need it. You can still see that there are different types of atoms in this structure. Okay. And this kind of representation works as you get to larger and larger objects as well. more complex stuff. In this day and age of cryo-EM becoming one of the dominant forms of structural biology, we're seeing more and more of these really amazing, huge structures that get very, very complicated once you start drawing pictures of them. So here's the figure from the journal article. To me, it's very difficult to see what is going on in the RNA polymerase up here, because they're using a lot of different colors, trying to show a lot of different things in one figure. And so in the molecule of the month, I simplified it immensely using different colors to show RNA polymerase, the two subunits of the ribosomes, and two transcription factors here that are tying the two together into one big expressome. I also wanted to show what was going on inside, and did that instead of trying to do it in one figure, do it as a separate figure, just doing a transparency here to show what's going on inside with the tRNA, mRNA, and the DNA. And again, the color scheme chosen to highlight those different functional pieces, a recognizable color for DNA, for RNA, for the tRNAs, for the RNA polymerase, and for the ribosomes. Excuse me, I'm going to lose my voice by the end of the week, guarantee. So here are a few examples of different pictures from different Molecule of the Month articles, just to underscore the fact that you guys have a lot of space for creativity in these different articles. The RNase P structure is going to have to deal with stuff like this, big complexes that have both RNA and proteins. In this one, I've colored the RNA and the DNA very recognizable colors, and all the different proteins similar shades of blue, just so you can see that those different protein chains are still there, but they're not all kinds of different wacky colors to make it distracting away from the thing that we're interested in, which is the nucleic acid. One of the teams is going to be working on binding of dexamethasone to its receptor. And so you might want to play games like this, where you're highlighting the structure of the drug itself and then where it is in the receptor. The team that's doing the designed folded proteins, they're probably going to want to create a figure that has multiple structures in one image, because there are actually 4 of those different structures. And they're probably also, since it's specifically related to protein folding, they're probably going to want to use ribbon diagrams like this one at the bottom. And this is a way I've been doing ribbon diagrams that show both the chain as a ribbon and also just a hint in the background to show that this is actually inside or a representation showing a larger protein that is composed of actual atoms. And then another team is going to be working on nanopores. And so you'll have to make decisions like, do you want to show it from the side? Do you want to show it from the top? And stuff like that. This one is actually a nuclear pore. Amazing that there are structures of whole nuclear pores these days. Quite remarkable. Okay, so those were the portraits that you'll use in the first 2 sections. We're also going to be putting together an interactive JSMOL in that exploring the structure section. And so in these, we're going to want to tell a structure function story, and we're going to have much, much more freedom with everything, with the representations, with colors, whatever you need to do to tell that story. We'll really press JMOL to its limits to tell these stories. And you might also want to do fun stuff like comparing multiple structures or showing close-ups of active sites or whatever you need to do to tell the story. And so here are a few examples of things I've done over the years. Here's a comparison of 2 different types of drugs bound to the surface of something. This must be the hepatitis target. showing, comparing, contrasting an early drug and then one of the drugs in much later development, refined after the structure was known. And then just here in slightly different colors in the background are the active site residues that are blocked by this inhibitor. So the story was actually talking about the details of what these drugs look like, the fact that this one is cyclic. Here's an example of talking about a whole protein complex, protein assembly. The interesting part was this signaling helix, which is the site of recognition between the cyclase and the G protein. So the JSMOL shows the whole complex and then highlights the things that are of interest. Here's an example of comparing binding of two different nucleotides to a designed form of an engineered form of the enzyme. They changed these two serines to acidic amino acids, which coordinates this might be a magnesium, and changes the specificity from NADPH to NADH. And then finally, for structures like this is probably going to be relevant to the RNase P topic that you guys do. The JSMOL might just be about wow factor. You know, just look at this amazing huge structure that structural biologists can solve these days, and giving people the ability to spin this around and look at the different parts. One of the big challenges of creating these pictures is that structural biologists will do whatever they need to do to get a structure. And so many proteins are flexible and difficult to study by crystallography or electron microscopy. And so crystallographers and microscopists, they typically cut them into pieces and solve structures of the pieces. And then as outreach authors, we have to figure out how to tell a story with a structure that only has a part of what we need to present, right? And so there are two approaches you can take to that. You can make up, model the parts of the structure that are missing, or you can use a schematic kind of representation to show that those pieces are missing, but they're not included in the structure. And so we do that latter thing most of the time in the Molecule of the Month. Since the article is so closely tied to the archive, we want to give people an idea of what they're going to see if they go into the archive and explore these structures themselves. So here's an example of an enzyme that was solved in two separate pieces with a little flexible linker missing, right? And so I chose to draw this as two separate pieces with some little dots here to show that a small linker is missing. For the coronavirus spike, the portion that ties it to the membrane was not resolved in any of these structures. And so I showed the membrane and these transmembrane segments just as a schematic representation here. It's enough to tell the story, right? Since most of the stuff that we're interested in and that was discussed in the article was in this the portion of the protein outside of the virus. But just to give people a hint that there's more to this story that isn't covered in the structure. And so I think there are a few examples in these 6 topics where you're going to have to deal with that. We'll talk about that in more detail as we get to the topics. Okay, so now, are there any questions about that before I move on?
David?
Yes.
Would you care to comment about scale in these representations where you're making models or including figures, just so that you can bring to light how big the complex is and where the important bits are, and how you deal with that? Like, you know, how do you make sure that things are at scale?
Scale is a tricky thing. So with the program, the illustration program that I use, I can set the scale explicitly. So there's a line in it that says, this is going to be 12 pixels per angstrom. So I know exactly what the scale is of those pictures, which is very important if I want to create a panel that has 10 different proteins, right? I want them all to be the same scale. So as in the topics we're going to do, as we're creating these pictures, we'll have complete control over the scale, so that if we are displaying two proteins that are very differently sized, they'll be scaled correctly relative to each other. The challenge is that most graphics programs that you use don't have easy ways to control that scale. And even worse than that, almost all graphics programs use perspective transformations. So things that are closer to you are bigger, and things that are further away get smaller. Makes it easier to understand the depth. But if you create multiple pictures, it really messes up that relative scale. So it's actually, with almost every graphics program in existence, it's difficult to create a figure that has multiple images, multiple proteins all drawn at the same scale. Yeah, so that's the answer to it, that you have to be very careful if you're using most graphic programs for that. That being said, most people don't do those kind of pictures. You know, they're not. It's only usually in an introductory figure in a textbook that people want to know the relative sizes of proteins. Another way to solve those kind of things, if you want to do it, is to create a file that has coordinates of both of the things, and you generate the picture in the same view in the viewer, which I guess you can do in Molstar. Yeah, something to pay attention to. And if any of the teams decide that they want to do a picture that compares multiple proteins, we can talk about ways to do that and make sure the scales are correct. Thanks for mentioning that, because that's a big aspect of everything that I do. I always try to draw things to scale. Any other questions or comments?
No, someone put a question in the chat.
Do I know how to get to the chat?
So David, the question is reading, do you use JSMol to add labels or use another program?
Good question. So my experience with JSMol and every other molecular graphics program in the world is that labels are awful.
Yeah. And
that they never come out right. So to me, the labels are useful when you're exploring a structure in one of these interactive programs to help you figure out what it is you're seeing. But for final images and final JMOLs and things, I always use Photoshop or something to make really nice labels. And those the schematic things I just showed, these schematic parts and these dots, they're all added in post in Photoshop. Yeah, so I can help with that. If you guys don't have Photoshop on your own computer or something similar, I can help with that kind of stuff to put the final things. But it's really the only way to get nice labels like these. Yeah, and if you're, some of you are thinking about careers in visualization stuff, and so Photoshop or Illustrator or something is going to be at the top of your list of things to do if you're not already doing it. Okay, are there other things in the chat? I unfortunately can't get to the chat from.
I'm happy to read the question out, the questions out. So there was actually another question from earlier in the morning. Natalie had asked about the teaser. So is this a one sentence abstract?
So Natalie, did we answer that question with those examples I showed? That it's
We answered that, no problem. Thank you.
It is kind of an abstract, but it's an abstract for an abstract to get people interested rather than just an abstract to summarize. Okay, so why don't we shift gears, and I'm going to do my little elevator pitch of all 6 topics, And so as I go through this, at the end, we're going to ask you to prioritize what you're most interested in doing, and we'll try to accommodate everybody in the topics that they want to do. Okay, so 6 different topics. You've seen this slide multiple times already, all with very different goals of what we're trying to do with them. So the first one is fetal hemoglobin. So the overarching question that you're going to answer on, or answer on this is, how is it that a mother can provide oxygen to a fetus, right? If they both had the same kind of hemoglobin, you'd never be able to transfer oxygen from the mother to the fetus. Okay, so that's the concept. There's also a really interesting secondary story about using gene therapy to express fetal hemoglobin in people with sickle cell disease as a way to bind oxygen more tightly. So that might be a nice secondary story to tell for this. The JMOL, you're going to take a deep dive into how it is that this fetal hemoglobin binds oxygen more tightly than the adult one. And this is going to be a nice one to work in because it's going to have a lot of appeal in classrooms. So this is one that's probably going to have a long shelf life and is going to be used for a long time. So it also uses this nice historical structure from Perutz from the dawn of structural biology. So that's number 1. Second one is this glucocorticoid receptor. So the big selling point of this one was that it's the target of dexamethasone, which is the drug that Trump was given when he caught it. So there's a story there to tell as being the only approved drug to fight COVID right now. And you'll be able to talk a little bit about these cytokine storms that are in the news.
That's not quite accurate, David.
Is there more than one now?
Remdesivir has been approved, right? Yeah.
Yeah.
But arguably this is the most impactful of the small molecule drug approvals,
Okay.
having the best impact in terms of outcomes.
Super. So that'll be your major story. There's also a really interesting story about how this receptor traffics between the cytoplasm and the nucleus, and there are chaperones that carry it back and forth as part of its function. That's interesting. And the JMOL will probably be comparing the drug bound to it to the natural ligand of the receptor, and those two are very similar. So you'll be able to talk about how this drug was designed based on the original, the natural ligand for the protein. Third topic is this ribonuclease P, which is one of 2 processive ribozymes in our cells. So ribozymes are RNAs that can do catalytic reactions. And so most ribozymes are simple ones that cleave themselves and mature themselves, but there are only 2 widely distributed ribozymes that can perform multiple catalytic reactions the way enzymes can do. This ribonuclease P, and does anybody know what the other one is? Quiz for the group. Silence, crickets. A molecule composed of RNA, mostly RNA, that can perform a chemical reaction using its RNA. Well, we'll leave that for the team that is going to do this. So it has an essential function in the maturation of tRNA. So it makes a clip in a precursor form of tRNA to form the final one. The secondary topic is probably going to be something about molecular evolution, the fact that there are different forms of this enzyme in basically all kingdoms of life have a form of this ribozyme, and then the JMOL is going to be one of these wow, just the structures are huge and amazing and wonderful and fun. This one, if you're particularly interested in the visualization of aspect of everything, this might be a good choice for you because it's going to be a real challenge creating an interpretable figure of this very complicated structure. Fourth topic is about citizen science and these a recent development where 4 different proteins were designed using this online tool Foldit. And so these are proteins that were designed de novo and ended up folding. Structure solved of those. And so the secondary topic will probably be talking about several other structures that are past successes of citizen science. Like there was an example of a protein structure being predicted by Foldit and then used to help solve the structure of that protein. And then I think the really fun part of this one is going to be the JMOL, because I think what you're going to be able to do is create a JMOL that with a bunch of buttons that allow you to turn on the hydrophobic core or the salt bridges or all the different things that that stabilize the structure of this small designed protein. So it'll be a nice way to create a teaching tool that shows all the different principles that stabilize protein folding. So, and I think that's going to be something that's going to be very useful in educational settings down the road. Fifth topic is looking at nanopores that are used for sequencing, rapidly sequencing DNA. This is one of the common ways of sequencing DNA these days. It turns out that the DNA slips through that pore and they can have it chunk through it one nucleotide at a time and sense what the nucleotides are. So the topic will talk about these rapid sequencing techniques that are widely used for DNA sequencing these days. Several different pore proteins are used or have been tested for this kind of biotech application. So the secondary topic will be talking about those different pores. And then the JMOL will probably look at an engineered version of this protein that sequences better than the natural version, and allowing people to explore the things that that are improved in that. And then the final topic is looking at something related to cancer. One of the major drugs used to treat cancer is cisplatin, which is a very simple drug composed of a platinum atom and 4 nitrogens around it. It binds to DNA and links 2 different base pairs next to each other, bends the DNA and blocks RNA polymerase, causes all kinds of problems and ends up killing the cell, killing the cancer cell. And so this is going to be part of our yearly theme on cancer next year. So it'll play an important role in that and be seen by a bunch of students as part of that. The primary topic will be just talking about this structure of cisplat, the lesion, cisplat lesion with DNA. The secondary topic will probably look at a few proteins that are blocked by this lesion and also a few proteins that repair it. There are some polymerases that can copy right past it. And then the JMOL probably will talk about this bend and how it makes a perfect site for proteins to bind that end up blocking DNA repair. So these cells are easier to kill because the normal processes of DNA repair are blocked. So that'll be a really interesting story to tell. So anyways, those are the 6 topics. Here they are: hemoglobin, glucocorticoid receptor, RNase polymerase, or RNase P, Foldit, design proteins, nanopores, and cisplatin, and cancer.